All right. Well, thank you very much. It's as Mary said, I'm David Gamm. I'm from the University of Wisconsin, and it's wonderful being up here in Canada and beautiful province and beautiful city. I've been here a few times, and I always enjoy it. And everybody's very hospitable. We also are having a sale on country leaders. So if you want to do a straight up swap, we're good with that. We know you guys have your own issues, but it's okay. So I'm going to talk to you about stem cell therapeutics for RP and AMD. I'm a pediatric ophthalmologist, so my practice is mostly... Oh, I hit that already. My goodness. There we go. So my practice is mostly dealing with retinitis pigmentosa, but my research bleeds over into a lot of AMD as well, and so I'm going to try and focus on both today.
Can we ask you to put the microphone a little closer to your mouth?
Okay, I will. And it will drift away. So then you just have to let me know. So also, there's a lot of slides here, and I know a lot of folks here have low vision. So at any point, if I fail to explain when I'm presenting on the screen, raise your hand. I'm happy to go back over it, and if there's something you don't quite get, because if you get too far in a talk and you kind of miss a couple of the concepts before that, it can lead you to get lost. So I don't want that to happen. I also want to disclose that I'm a co-founder and chief scientific officer for Opsis Therapeutics, which is a company that's deriving retinal cells for therapeutic uses using induced pluripotent stem cells, which is what I'll talk about. And I consult for Fujifilm. However, I'm also a very skinny guy, so I'm thinking of a little side venture. So next year I'm going to have a booth, I'm just telling you. So this whole stem cell thing just falls through. I think I got it. So, all right. So just to give you a little bit of background, this is kind of a stem cells 101. This is what I go over with my residents and students. So if you think just in broad concepts, I'm not going to focus in on retina right away. I'm just going to talk to you about stem cells and what are they. Because one size does not fit all. People throw that term around all the time, and they do it usually to try and coerce people into spending money they shouldn't spend. But I'll try and give you a little bit of knowledge on that. So here's I'm showing you a healthy cell. So this is just a kind of a nice, smooth, nice cell. And on occasion, if we're unfortunate, these cells make up our entire body, right? There's trillions of them. But if that cell gets sick, it's a disease cell. Most of the time, if that happens, let's say it's an infection, we take an antibiotic, we have some sort of therapeutic that's available to us, and we can revert that disease cell back to a healthy cell. So if you get a cold and you're not feeling well, it doesn't mean that your throat's going to degenerate and go away. Your body will take care of it, or you'll have a therapeutic. But unfortunately, there are diseases that are called degenerative diseases, wherein those cells don't just get sick, but they actually degenerate, they go away. They break up, they die. And in that case, there's really no way to put Humpty Dumpty back together again. It's not going to become healthy anymore. So in those situations, if you're lucky, what'll happen in some tissues is that there's a resident cell that's kind of hidden within that tissue. So let's say it's your liver, let's say it's the lining of your gut, or your skin, or your blood cells, because your skin is always kind of sloughing off, and then your body knows how to make more skin. So in those cases, there are these resident tissue stem cells. So these little cells that just sit there kind of quiescent, they don't do much until there's an injury, and then they're induced to make, wrong way, and then to make more of themselves and repopulate the cells in that part of your body that have been lost in the course of disease or injury. Unfortunately, the retina is not one of these tissues, so it does not have a capacity to repair itself once it's been damaged. And the same goes for your central nervous system. So that's why things like Alzheimer's, Parkinson's disease, Lou Gehrig's disease, and macular degeneration and RP continue to plague humankind because the body can't replace those parts themselves. So what's your options then if that happens? So if you've undergone a significant amount of degeneration, then and your body can't replace it, and there's no kind of treatment to do that. There's two things that you can do. You can either bypass it. You can say, well, the heck with that cell. Let's try and get other cells that are still around to do the job that the cell that just died is supposed to do. That's called bypass, and those are your prosthetic chips, optogenetics, things like that. Or the other possibility is to try to replace them, and that's really what I'm going to talk to you today about with regard to stem cells. Although stem cells have applications for everything else as well as a model system. So we can make these cells and these tissues from any person on the face of the earth, and we can test almost anything with them. So there's applications beyond just cell replacement, but I'm going to concentrate on the replacement aspect of things. Okay, so what types of cells are we hoping to replace? So you heard already from Dr. Merker the background on what your retina is. So it's the film in the back of the camera being the eye. If you pull it out, it looks kind of like a thick, somewhat opaque-ish looking cellophane, and that lines the back of your eye like a wallpaper. But if you look at it really carefully, it's actually a very elegant layer cake of different types of cells. So it's not just one amorphous, sheet, but it has specific cell types that are arranged in a particular order, like a tiny circuit. And deepest within that circuit are the photoreceptors in the retinal pigment epithelium, and they're the Batman and Robin of vision. That's where vision starts. Light passes through the eye, it hits the photoreceptors. The RPE, the retinal pigment epithelium, are the helper cells that make sure that the photoreceptors work properly. The photoreceptors are all divas. They die without the RPE. The RPE doesn't get any credit, but without it, the photoreceptors wouldn't work. And then once the photoreceptors, the rods and the cones, detect light and are stimulated, they pass that signal up through the retina, eventually to the ganglion cells, which are affected in glaucoma. They send their signal back to the brain, and your brain does the seeing. So which of all these different cell types are we really focusing on early on with regard to replacement? And those two cell types are the photoreceptors and the RPE. So that kind of that Batman and Robin that I mentioned earlier. And so RPE, as I mentioned before, are the helper cells that keep the photoreceptors working well. Okay, so then I'm going to focus just in on that. So the other cell types we can talk about later, but there are reasons why they're harder. They're further down the road in terms of trying to replace them. And of course, we have the whole, what if we can replace the whole retina in cases of really severe degeneration? And we could talk about that. But that's really, there's a spectrum of things that we can approach, and it's all hard, but there's hard and really, really hard. And so let's just start with the hard stuff. So here's an example now of what pictures of cone cells. This is what cone cells look like. They kind of look like long, thin cells that have these little proboscis that stick out that are called outer segments, and they do the capturing of light. And those proboscis interdigitate with the RPE, which are feeding them, and that's shown here down below. And so the diseases that we're interested in are those that cause either primary degeneration of the photoreceptors, so the rods and or the cones, directly, and a lot of that's retinitis pigmentosa, or diseases that cause degeneration of the RPE, that helper cell, which, if that happens, the photoreceptors, being divas, can't stick around, so they die as well. So they die a secondary death. And that's macular degeneration. So that primarily affects the retinal pigment epithelium, but you don't go blind because the RPE dies. You go blind because the photoreceptors stop functioning because the RPE died. All right, so the nice thing about stem cells in general is that you hear a lot about gene therapies, and they're wonderful, and they're coming down the pike, and there's going to be more of them for more people. But they're specific for the individual gene defect for retinitis pigmentosa. And also there are diseases that really aren't due to one single gene that you can just fix, like macular degeneration. So with stem cells, it's relatively agnostic to what caused the death of your cells to begin with. So whether it's gene A or gene Z for retinitis pigmentosa, or whether it's AMD, it doesn't really matter. It does, that's a very broad stroke, but what matters more is what stage you are in the course of that disease. Too early, you shouldn't be getting cells replaced because your cells are okay. We should be trying to protect them. It's better to keep what you've got than to replace what you've lost. Way, way too late, and the whole retina could be screwed up, in which case one cell or two cells isn't going to help you either. So you kind of have a window there. But our hope is that we can apply this to a much broader group than, say, we could with gene therapy. And so, as I said, the candidacy will vary based upon the condition of the retina, and there's some examples up here, a whole host of different types of degenerative diseases caused by genetics, or primary genetics, and then things like macular degeneration, maybe even chronic retinal detachment, or injuries. Blast injuries is something that we're working with the Department of Defense. So, where do these spare parts come from? Essentially, that's what we're talking about. We're talking about a two-step process. We got to manufacture the spare parts, which would be the retinal cells, the photoreceptors, and the RPE, and then we got to install them. Very simple. Make, install. So first of all, if there's nothing to install, that doesn't matter. So along came human pluripotent stem cells in the late 1990s, and it was brought on board by Jamie Thompson at the University of Wisconsin, and I was very fortunate to be a resident and a fellow there at that time, which allowed me to learn quite a bit about it at an early stage. And at that time, what we had were embryonic stem cells, so human embryonic stem cells. And they were derived from in vitro fertilization clinics where couples who had had their kids and were going to destroy the embryos anyway, the folks there at the University of Wisconsin used those cells in the inner cell mass to create this human embryonic stem cell system. And they created 22, and George Bush said, that's all you can do, just do those, and you can use those, but that's that. And in actuality, the fact that the people were restricted in making lots and lots of different lines helped us at an early stage because it forced us all to use the same football, or soccer ball, whichever. And so instead of us all having 300 different types of tools, all of which were slightly different, we all were using the same thing. And it got us to be on the same page and learn a lot about the system that I think otherwise could have gotten really messy really quick. But then in 2006, 2007, Jamie Thompson, as well as Yamanaka in Japan, found a way to take adult cells, skin at first, but also now what we use mostly now are blood samples, like you might submit for a cholesterol check, and then we take certain blood types called T cells or B cells out of those, and we could reprogram them genetically back to a state that's essentially the same as an embryonic stem cell. So we have now a source that can be derived from any human, any person walking. Well, you can actually do it from dogs and other animals too, but where we can create a stem cell from any human walking the face of the earth. That's interesting if you've got a particular disease that we want to study, because then we can make a model of your retina in a dish and find out why those cells died and maybe develop a treatment for it. But also perhaps use those cells to go back into a patient. And to replace the parts that have been lost. So what we do in my lab now is 99+% is with induced pluripotent stem cells. Now there are other clinical trials that are going on right now. Reneuron and JSITE are using fetal retinas. You say, okay, where else could you possibly get human retinal cells? Well, it's possible you could get them from fetal tissue as well, and there are companies that are doing that now. As you might imagine, that's not renewable. You have to go back and get more. Also, those are not stem cells. Those are cells that have a limited capacity to make the cell types in our retina. They've been used before, and they really have never been shown to be bona fide photoreceptors or RPE. Well, the RPE, yes, but in terms of photoreceptors, no one's really ever shown that they've been able to cultivate them in culture and make real true photoreceptors. And then what about human adults? What if you just got a cadaveric eye? So somebody passes away, they donate the eye, which typically these days is to remove the cornea for folks that have corneal disease. But what if you were able to harvest cells from their retina? That has also not been successful, in large part because those cells are very, very mature, and pulling them out and manipulating them, they often die. And it's again, a limited source. You think about, right now we have deficits in getting liver transplants and kidney transplants. The benefit of pluripotent stem cells is, as I'll show you, they're bona fide, authentic sources of your retinal cells, and it's an unlimited supply. Once you make one of these lines, you can propagate it forever. And so it's an unlimited, we can make billions and billions of cells from a single cell line. So how do we do that? Well, we actually recapitulate developmental biology. So it's not magic. These pluripotent stem cells, when they first start, have the capacity to make every single cell type in your body, and they will. That's a kind of a cool thing, but it's also a very difficult thing when you want it to make one specific part. You don't want it to make all the parts in the body, you just want to make it one or two. And you want it to make it very reproducible, very pure. You want to be able to pull it out. You want to make enough of them where you can treat a lot of folks. So that's where the science comes in, where you apply developmental biology principles that have been learned over decades and decades from very smart people. And we try and get or coerce these cells to go down a particular path. In my case, it's towards brain, and then veer off to the right a little bit towards eye, and then make photoreceptors or RPE. We do that using various chemicals, all of which are safe ultimately to be put into humans, so the chemicals themselves don't carry over into the humans, but we have to make sure that every step of this process is okay to ultimately take that product and put it back into a patient. We also use physical forces and other cues. And here's ultimately what we get. This is some retinal pigment epithelium I'm showing here. They're pigmented, they're kind of brownish black, they look like little stop signs, they form a nice sheet, and they can do all the tricks that a normal human retinal pigment epithelial cell can do. They turn out to be, that's not so hard. So RPE, that helper cell layer, is one of the easiest things to make from human pluripotent stem cells, ES or IPS cells. Because of that reason, it's really led the way in terms of utilization in patients. And so most, and I'll talk towards the end, some of the clinical trials that are going on, they're all at this point using retinal pigment epithelium, not photoreceptors, because it's easier to make and easier to make in a way that's palatable to the FDA or the authorities that are tasked with making sure things will ultimately be safe when they're used in humans. Also, the RPE themselves are not neurons. They don't have to fit in or synapse or make an electrical connection with another cell. It's a helper cell. It kind of just has to sit there and do its job, which makes it a little easier from an installation standpoint. Whereas photoreceptors, they have to interdigitate or work with that helper cell, but they also have to make a handshake with the next set of cells within the retina to relay their signal. So neurons have to do that. That's a harder thing to do. So what about photoreceptors? Not only is our RPE easier to make in terms of just looking at them, finding them, removing them, and making more of them in a dish, but they grow by themselves. So they grow, once you isolate them, you can make a lot of them and they grow happily just by themselves. Photoreceptors, because of developmental biology, don't do that. They grow within a community of cells that will ultimately make up the whole retina. So for that reason, when we grow these little tiny retinas, we're not just growing photoreceptors, we're growing all the different elements of the retina. We essentially make mini retinas in a dish. And so that's, again, very interesting from a scientific standpoint, but we have to somehow extract the cells, those photoreceptor cells, if we're going to use them for therapeutic purposes. So here are the tiny little retinas. They look like golden Cheerios. And I won't go through, it took a decade or more and a lot of patents for us to be able to figure out how to do this. But they essentially recapitulate the developmental steps necessary to make a retina. So we start with these very tiny little guys that are just like the early onlage of your retina when you're in your mother's womb. It kind of bulges out and initially is comprised of only one type of cell. And then over time, each one of the different cell types gets born in a particular order and in a particular space within those tiny little golden Cheerios, until ultimately you have something that has essentially what we would look for a late third trimester human retina, where you have cone photoreceptors that detect color and high acuity vision. Beneath them, we have a lot of rods that do the night vision and peripheral vision, and the orientation is very similar to what we would see in a regular human retina. And if we compare that to, say, for instance, a monkey retina, you could tell the difference. It's definitely not perfect, but it's pretty close. And the cell types themselves can do all of the different things that we would expect a cell type that's that mature, which is still relatively immature, to do. So just a couple of little videos here. On the left, what I'm doing is spinning around just the surface of one of these retinal tissues that we've made in a dish. And there are thousands of these that we'll make at one given time. And we can do them in vats now where we'll have on the order of 1 billion or more of these cells growing at one time. And so when green, I'm showing you the rods, in red, I'm showing you the cones. And if we look very, very closely, so if we zoom up on those now, I wanted to do the zoom. There we go. And if we look much, much closely at what those little green and red dots are, they have all the attributes of a cone or a rod. And not only that, I'm really just spinning pretty colored things is what I'm doing. So take my word for it for the rest of the stuff. But they have these, remember I mentioned before this little proboscis, those little antennae that the photoreceptors have to have in order to detect photons of light and initiate vision? Well, they have those too. So these little guys at the very tips of all these little photoreceptors have these little outer segments, and they're chock full of the appropriate subparts necessary to detect light. And when we test them to see whether or not they do that, they do so, and they detect light at the proper wavelength. So a blue cone detects blue light, and a green cone detects green light, and a red cone detects red light, and a rod detects isn't color sensitive whatsoever and has a different wavelength that stimulates it. So once we knew over the course of many, many years that we had something that really looked like a duck, quacked like a duck, flew like a duck, and we thought it was really a true spare part that could help patients. The next question is, can we convert all those steps that we have to do in a laboratory, which aren't safe to go into a human? We use a lot of things that wouldn't be safe, but can we work with the folks at the University of Wisconsin whose job it is to convert laboratory-based discoveries into human therapies, can we convert that? And so we work with the Weissman biomanufacturing facility that's luckily just a couple floors down from my laboratory. And over the course of a year or so, we were able to do that. And we're able to pull out all those photoreceptors, again, in very high quantity, very high purity, freeze them down, thaw them back up, all the sort of manipulation stuff you have to do just to make it a viable treatment that you can transport across countries. And this is kind of what they look like when they're very young, just, again, some pretty colored dots. And then the idea now is once you have the spare parts, which we do, so we can make the RPE, we can make the photoreceptors, the cell types that are lost in AMD, the cell types that are lost in retinitis pigmentosa, and some injuries as well. How about getting them to the right spot? Can we do that? Well, thankfully, that's already being done, and for the most part, we have to do this by going under the retina, because as you remember from the very beginning slide that I showed you, that photoreceptor and those RPE are deep within the retina. So you have to be able to make a small hole in the retina, lift the retina up, essentially form a detachment of the retina, and then place the cells in where they would normally be that you're trying to replace. And here's an example. In red are all of the new human photoreceptors that we've made, and we've placed them into a rat that's blind because it's lost all of its own photoreceptors, and we can recapitulate that layer of cells, and they will form the correct handshake necessary. But the simplest way to do that is just to kind of squirt them in there. And while that is the simplest way to do it and kind of probably the first wave of how we'll deliver these cells, it's almost assuredly not the smartest and best way to do it. Just like every other technology, you didn't start with airplanes that could fly across the Atlantic. You started with folks trying to glide off a hill and then getting back up and going farther and farther. That's the same thing with this technology. It's not going to be a grand slam home run right out of the gate. It's going to take iterations and versions 16.0 and 18.0. And so we're already kind of working on that. And so one of the things we want to do is take those cells and not just randomly put them in that space that's been vacated by disease, but rather orient them in a particular way. And so we make these very small fabricated scaffolds that we can seed the cells into so that they have an orientation in them when we're putting them into the eyes. It also helps us to delineate where we're going to deliver those cells and to provide a specific dose so that we can follow these various things and get better information back out of the patients once we're doing these trials. Okay, so whose stem cells do we use? I said before, we can make it from anybody that walks the face of the earth. So do we want to do that? Do we want to make them specifically for each and every person that needs them? And that can be done. It's called autologous stem cell therapies, but it's exceedingly expensive. So outside of Bill Gates and a few other people, that's probably not a viable thing that we can do, and it would really crush the medical system. But we have other options. One would be a one-size-fits-all, and we can manipulate these cells such that perhaps we wouldn't even recognize them and we wouldn't reject them, even though they're not from our own personal blood or skin. We can also partially match, so we can develop a bank of cells like you would match for a kidney or a liver transplant. Not perfect, but closer than a random cell type. And then, as I said before, there's this idea of a universal cell that we can manipulate that can evade the immune system, perhaps, but there's dangers to that too. Okay, so I'm going to give you a brief kind of stem cell clinic update. How am I doing on time, Mary? Doing all right? Because I want to leave time for questions too. Anything with just kind of the general background about stem cells and making retinal cells from them? Anybody want me to push on? I will push on. Push on. Okay, so I'm going to give you a little sense of where we are right now. I've already told you the photoreceptor end of things, which is what we're doing right now, is about 2 to 3 years away from the initial clinical trials. And that's because it's so hard to make that cell type, as for the reasons I told you earlier. But the retinal pigment epithelium is well on its way in clinical trials and being tested in patients. And here's just an example of some companies that are in this space. And some of the trials that have been done, some of the early ones were just taking the retinal pigment epithelium and not making the sheet of cells, but just squirting those cells in. And they showed that there was no tumors, there were no untoward effects for the most part, but there was really not much in terms of a benefit to patients. But then a couple of groups, one in Japan and a couple in California, one in London, started prefabricating the RPE onto sheets and placing those into patients who had macular degeneration. These are late stage. These are very late stage macular degeneration, so not the early forms. And they were able to show, again, that it was safe, that they could get the cells in the right place, they could be installed, in other words. And it seems to slow down the process and perhaps improve a little bit. But as I told you at the beginning, the reason you lose vision in macular degeneration is not that you lose the RPE, the helper cell, but because you lose the photoreceptors. So if you're just replacing the helper cell, really the goal is to slow things down, not necessarily to bring things back. And to that degree, to that extent, it seems to be working. And here's just a couple of examples of these very tiny little implants where they grow. These are all ES cell or embryonic stem cell derived RPE that they're growing on these little sheets that are then implanted into the eye. Okay, and this is from the London project to cure blindness. It looks kind of similar. Actually, Pete Coffey, who's the head of the London project, and Dennis Clegg, who's the head of the California project to cure blindness, they're good buddies. They actually both work at the University of California in Santa Barbara, and they're hilarious to be around because they're doing the exact same thing, but they have their scaffold. One's made of polyester and one's made of parylene. I don't know the difference between the two, but talking to the two of them, you'd think that it was Mars and Venus, but they're both essentially getting a similar effect. Okay, I mentioned before, there's J-Site, there's Reneuron, they're using fetal cells, and they're getting some interesting data too. I can't quite understand it. It's hard to explain how they're getting their results, but they have some intriguing results from those two. And then what's coming soon, Opsis Therapeutics is the company we started, and we're looking at 2 to 3 years, as I mentioned before, to first get these into patients. And there are other groups trying to do the similar thing, which is great. You want as many shots on goal as you possibly can. And then, of course, I have to mention the fact that the Fighting Blindness Canada has been wonderful to me. I'm part of their SAB. They invited me to be on the SAB. I jumped at the chance. It's a great group. I really enjoy working with organizations that have a real focus on patients. And so we were the recent recipient of a Restore Vision 2020 initiative grant, which is a wonderful opportunity for us, along with our colleagues at the University of Pennsylvania. And the company, Opsis Therapeutics, to take a lot of what I told you here and to perform critical experiments necessary to bring this to clinical trial. So that 2 to 3 year window, that would be longer if it wasn't for the Fighting Blindness Canada. So I thank everybody for that. So to summarize, we have the technology now. We can make human retinal cells in an unlimited supply and whole tissues, as a matter of fact, in a laboratory dish. Installing these parts remains a very significant challenge, and that's going to require time and very rigorous studies in humans and in animals to really understand what these cells are capable of doing and what they're not capable of doing and how we have to further manipulate them to improve outcomes. The eye is just a major target for stem cells in general because there's accessibility. God gave you 2, you can play around with one. So that's, you only have one brain. It's a little higher risk there. Existing trials are in their early stages, but we're starting to get really important data out of humans. Thus far, it's only been with retinal pigment epithelium, using both the embryonic stem cells and iPS cells. And although they're not truly stem cells, there are trials that are using human fetal cells as well. And the photoreceptor replacement and the combination of replacing photoreceptors and RPE, which ultimately is what folks with end-stage macular degeneration need, are on the horizon. So last 2 slides, and these are perhaps the most important, and I alluded to this earlier, and that's establishing the idea of realistic expectations. Okay, so we're not going to go from, here's a cell a source of replacement cells for your retina, to giving you your license back, or to reading the New York Times if you have AMD. It is going to take time. It is going to take thoughtful process and working together in collaborative forums and getting feedback from these early clinical trials to make really meaningful improvements in patients' vision. So I just want folks to know that. It's a marathon, and right now we're still in the crawling stage. And then the final word of caution is that all stem cells are not created equal. People love to use that word to bilk you out of your money. It's not a panacea. It's not going to cure everything that ails you. It's a real science, and it has to be applied in a very thoughtful manner. So there's limitations in terms of what it can do, and there are times when it's the last thing you want to do. And then be wary of anybody that tries to sell you something that says stem cell. I mean, it's all over the place. They're in Madison now. It's all over the United States. I'm sure it's here in Canada too. They'll rub it on your face, make you young again, all this sort of stuff. If it's too good to be true, it is. Listen to your mother, listen to your grandmother. They make these far-reaching claims, and essentially, if they're asking for money, it's not legitimate, because we're not at a stage at this point where anybody should be making money off of this. This is where we pay you to enroll in a study. You should not be paying us. If somebody's asking you for cash, they have a conflict of interest, and they're doing something they shouldn't be doing out of ignorance or out of greed. And so if you want, I actually put together kind of a top 10 things about what to be wary of for stem cell clinics. It's available. I direct the McPherson Eye Research Institute at the University of Wisconsin. It's freely available. So at www.vision.wis.edu, I have kind of a longer essay, as you can probably figure out. I like to talk. So I have this long essay about what the retina is, what stem cells are, a lot of kind of a mini thing of what I just gave you today. And then my associate director looked at me and said, no one's ever going to read that, Dave. Make a top 10 list. And so I did that. And it can be downloaded and free to be used and no restrictions. Anyone can use it anytime. So again, thank you very much, especially to this group, and I appreciate the opportunity to talk.
Dr. Gant?
Yeah.
You talked about creation of the stem cells, you talked about delivery method, but what about future degradation? Like if you're able to, in particular I'm interested in the photoreceptors, if you're able to implant and replace the photoreceptors, what's the long-term prognosis of those photoreceptors, like maintaining their integrity?
Okay, so great question. It goes back also to the idea of whose stem cells do you use, right? Because if I take them from a person who has retinitis pigmentosa, those same genes that person has will be contained in those stem cells, right? So if you're going to do that, you have to do what's called gene editing or base editing, and you have to correct the gene defect, which adds more time, more money, and a higher FDA hurdle to get it back into patients, right? So that's autologous. So if it's going to go from you back into you, then you have to make those changes. That's why that's not our approach, right? So that's too expensive, too time consuming. Our approach is to use normal or wild type and to use either one universal line that can go into anybody or a bank of lines that would be for a subset of communities. And surprisingly, you can cover the U.S. population with about 95% of the population in about 80 lines. Still exceedingly expensive, but you can. It's reasonable to do that. And then we have these other ideas, these universal cell lines that can evade immune systems. So that's a one-size-fits-all. In that case, we would have to really look at the genome of that cell line to make sure that it doesn't have anything untoward, but it wouldn't have those things. Okay, so that's the first part. The second part then is the rejection aspect of it. Theoretically, it would last forever, or as long as a normal photoreceptor would last. And since their age at the time we make them is birth, right? So normally our photoreceptors should be around for 90, 100 years. So theoretically, they should be fine. But you're putting those new cells into a diseased retina, right? They're not perfect retinas. So you have perhaps a toxic environment there. It's a new wine into old wineskin sort of a metaphor. So we don't know. And that's why I talked about the time window. There may be certain diseases or certain stages of disease where you're putting that brand new, healthy, ready to go cell, and it will just succumb to whatever bad environment there is there. So the initial studies will be select in patients where we hope, based upon imaging and other techniques that we can use, where we're minimizing that. And then as we go forward, some of the improvements in the technology won't be in the cell manufacturer or even the installation, but how do we prime the host retina to accept the new cells? So how do we kind of clean up the garbage, as we heard about before, so that they'll be able to behave more normally once they're there? And then there's the rejection part of it. And like I talked about before, it's possible that all that stuff works out great, but then your body says, well, that's not me, and destroys it. So those are all things that could happen. So the duration of the treatment is something we won't know until we get it into patients. Great question. Ma'am in pink?
This is one last question. Thank you so much for explaining that. That was so easy to follow. I really appreciate that. Also, I've never heard George Bush praised in a lecture. But my other question
I'll take him back.
no, it actually worked out the way you explained it.
The ES cell thing. Yes. Yeah, no, it's actually a different perspective I have on that. But it's one of those things where you look back 10 years and you go, you know what? It was probably a good thing.
Yeah.
Things would have gotten crazy.
Yeah,
and
exactly.
we now know that there's a lot of differences from one line to the next. And if we had 300 different lines and everybody had their own specific line in their own lab, we would have been getting all kinds of crazy off-the-wall sort of findings, thinking that that was the real thing. And it turns out we just didn't understand the variabilities that there were in the system. So making us all use the same stuff at the very beginning was, it wasn't intended to be that.
Well, in retrospect.
Yeah,
sure.
right. In retrospect was probably a good thing.
My quick question, because I know everyone wants to get to the break. You said the eye doesn't have the stem cells.
Correct.
Any other body parts that don't have them?
Well, I would say most don't have them that have the capacity for really what we would think about for regeneration. So the ones that do for sure are your skin and the lining of your gut, your blood, because your red blood cells only last 3 months, and so your body has to continuously replace that. And if you can't, then it's a disease called aplastic anemia. But the prime example are neurons, so your central nervous system. But there's limited capacities perhaps, and I shouldn't be so. Whenever anyone stands up and tells you something that sounds really dogmatic, they're probably wrong. So there's always shades of gray. The tissues that we have probably have limited capacities to restore certain things. But for instance, we can't take out a lobe of our lung and it will grow back. In some animals, that'll happen. You can do that. And the eye will do that. So some fish, some urideals, some salamanders, you can rip out their retina, they'll grow another one. But they also don't have nearly the complexity of the systems that we have. And so it's kind of a give and a take, right? When you start building complexity into anything, the ability to recapitulate that in all of its complexity becomes more and more difficult. And so your simpler organs, your simpler kind of processes, they tend to be able to restore themselves. But the more complex organs, more complex functions, we don't have that. Yeah.
Okay, thank you again,
Thank you.
Dr. Gamm.